In the journey of protecting our pollinator species, understanding the risks they face from pesticide exposure is very important. A crucial aspect of this assessment is the measurement of pesticide residues within the plant resources that pollinators rely on. One prominent route of exposure by which pollinators encounter pesticides is through nectar, which is used primarily as a source of sugars for energy. To comprehensively measure the extent of pesticide exposure, it's necessary to analyze pesticide residues present in nectar. The first step in the process is sampling our resource of interest, the nectar. While there are several methods available for nectar sampling, we focus on the microcapillary tube technique. First, it's essential to work in a clean environment. Start by covering the work surface with clean paper and wear a new pair of gloves. By changing both between each different plant, we prevent cross-contamination between samples. In this case, we are working with salvia. Carefully cut the flower spikes adorned with florets from the plant and delicately remove each floret from the spike. Now we are ready to sample nectar from the separated florets using a glass microcapillary tube. To access the nectar, gently squeeze the base of each floret, applying pressure from the body of the corolla towards the point where it was attached to the spike. This simple yet precise technique pushes the nectar out where it's accessible for sampling. Repeat this process with as many florets as you need. Note that the volume of nectar collected depends on your specific requirements. One approach is to continue collecting nectar until the microcapillary is full, ensuring that the volume collected matches the tube's volume. In some cases where there is not enough nectar to fill the microcapillary, you have the option to estimate the volume by measuring the length of the nectar within the tube and then calculating the volume based on proportionalities. Once we have collected our nectar sample within the microcapillary tube, the next step is to transfer it into a microcentrifuge tube. Insert the microcapillary tube into the microcentrifuge tube. Next, attach a micropipette valve to the upper end of the microcapillary tube and squeeze the pipette valve gently but firmly to push the nectar out. Be careful not to draw the nectar sample up into the micropipette valve by applying continuous pressure on the valve until the nectar has been ejected. With our nectar sample transfer, the last step is to preserve it by storing it in a freezer until analysis. This will help prevent degradation of the pesticides. Join us next as we continue this exploration with exciting and essential details related to pesticide analysis in nectar and other plant parts. Our goals are to better understand how pest management practices influence contamination of floral resources, how pesticides impact pollinators and the environment, and to identify safe management practices that reduce risks to pollinators and the environment.